genes associated with diseases to better understand the molecular architectures and pathophysiology of genetic disorders. Claudia believes that the application and understanding of human genetics and genomics can lead to improved treatments and the realization of precision genomic medicine, because the beginning of precision medicine is an accurate genetic diagnosis. Claudia has dedicated a great part of her career to the study and identification of novel genes responsible for rare genetic disorders to provide molecular answers to patients in undiagnosed diseases. The people who join us on Zoom, we kindly ask you to write the questions down in the chat so the speaker can answer them in the corresponding section of the talk. We kindly ask you to put your mobile devices on silent for the duration of the conference. Thank you very much and welcome.
Hello, everyone. Thank you for the invitation to participate in the STEM week. I would have liked to be with you there, but some unforeseen circumstances prevented that, but I'm happy to be with you remotely. So today I'm going to talk to you about genomics for health equity in Latin America. So my research primarily focuses on Mendelian diseases and rare diseases. So I'll start by giving you a landscape of what are rare diseases and how do we consider these diseases in Mexico and Latin America. So rare diseases, and this is very timely since just yesterday we had Rare Disease Day around the world. So rare diseases are defined based on their low prevalence in the general population, meaning that they occur in very low frequency and affect a small number of individuals at a time. This prevalence varies between different countries and in different regions. So in the US, for example, a rare disease is one that affects less than 200,000 people, according to the definition of the Orphan Drug Act of 1983. In Europe, it is a condition that affects 1 in 2,000 people. And in the Latin American and Caribbean region that encompasses 33 countries, there's no consistent definition throughout the region about what a rare disease is. Some countries like Argentina, Chile, Mexico, Panama, and Uruguay have the definition of less than 5 in 10,000 people, but this definition is also variable in, for example, Brazil or Colombia. Also, there are many countries that do not have a definition of what a rare disease is. And this is relevant because this definition marks whether the health system recognizes some of these conditions and the patients that are affected by these conditions and whether they have access to healthcare services and other social services. Also, the number of rare diseases that are recognized by different countries is very variable. So, for example, in Mexico, until May of last year, 2023, only 20 diseases were recognized. And in July of 2023, the General Council of the Health Ministry in Mexico recognized 5,500 diseases that are part of the catalog of the World Health Organization. Individually, they are rare, as I mentioned, in terms of how many people they affect at that time. But as a group, they can be very common. So as a group altogether, since there are more than 7,000 conditions that have been described as rare diseases, they affect about 6 to 8% of the world population. In the Latin American and Caribbean region, we estimate that there are about 50 million people that are affected by a rare disease. And in Mexico, only we estimate that there are 10 million Mexicans that live with a rare condition. Also, this is just the patients. Rare diseases affect not only the patients that have the condition, but also the caregivers and the family that surrounds them, and also broadly the society in which they live. So this is a healthcare problem that is of interest to the whole society. Additionally, in the region of Latin America, we have about 50 million indigenous people. And based on these prevalence estimates, we estimate that there are about 3 to 4 million people, indigenous people living with a rare disease. And this is also important because we know that most indigenous people are part of vulnerable populations that have low access to healthcare services. And these conditions can present with a real burden for their health and for their economics in these populations. Also, as I mentioned, there are more than 7,000 conditions that have been described. We think there are about 10,000 rare diseases, and 80% of them are genetic. However, the molecular cause of more than 6,700 diseases has been defined. So that means that we can actually diagnose these conditions using molecular technologies that interrogate the DNA of the patients with these disorders and find an accurate molecular diagnosis. Unfortunately, only 5% of rare diseases have an available treatment. So this also makes for a huge unmet medical need for these patients. In 1990, the Human Genome Project was launched, and it was finished in 2003. And the main accomplishment of the project was the production of the first human genome reference sequence that most people are aware of and are familiar. However, one of the additional goals as part of the Human Genome Project program, and also part of the benefits of the Human Genome Project, was the development of new sequencing technologies and the decreasing cost in sequencing of DNA broadly, but specifically in this case of human genomes. So as you can see here, the Human Genome Project costed about $2 billion to be produced. But then since 2001, when the draft of the Human Genome Project of the reference sequence was released, we've seen a steep decline in the cost of sequencing. And nowadays, this cost has been reduced very prominently. However, after the Human Genome Project was finished, there was a lot of debate on whether all that was invested, all those $2 billion that were invested in doing this project and producing the reference sequence, and all the hype that was generated by the Human Genome Project would actually pay off and would actually be beneficial for patients living with genetic diseases, and more broadly to humankind to advance science and biology and medicine. So around 2007, 2008, there was this question about whether sequencing would become just a technology of commodity, where only the rich and famous would be able to afford paying for sequencing their whole genome. And most of these people really didn't have a medical need that needed to be addressed. So the question about whether this was going to serve to study genetic diseases was very much a hot topic at the time. So around this time, we started this project that led to the first proof of concept that we can utilize whole genome sequencing of a patient with an undiagnosed genetic disorder to identify the molecular cause of the disease. So in this case, we sequenced the whole genome of Dr. James Lobsky, who was my PhD advisor. And I did the analysis as a PhD student for these whole genome sequence data to identify the variants that were responsible for the disease in him and his siblings. And so this was, in this way, we demonstrated that we were able to identify from the millions of variants that you can find in a whole genome, the actual disease-causing variants associated with a genetic disease. So since then, the implementation of genomic sequencing technologies to the research and later the diagnosis of genetic conditions has really revolutionized how we approach genetic diseases, and more specifically for this particular talk, the rare diseases. So since 1986, when the first genes associated with genetic conditions were described and published in the literature, so you can see here in orange the number of genes per year that had been published utilizing conventional methods, meaning linkage studies and large pedigrees that were needed to identify these genes associated with genetic conditions. However, in 2010, when we published the study that I just mentioned, and other groups started publishing new developments on, for example, capture technologies to perform exome sequencing rather than whole genome sequencing. So you can see here the contribution of the new next generation sequencing technologies to the identification of novel genes associated with genetic conditions. So this has really changed in the last decade how we study genetic diseases, especially for conditions that previously had eluded these conventional methods of studying genetic diseases, such as linkage analysis, as I mentioned, because they are disorders that occur, as we call it, sporadically. So there's no family history of a genetic disease, and then these patients happen to have a genetic condition, and these are generally caused by de novo mutations, meaning mutations that occur either in the germline of one of the parents or very early on during the embryonic development of the patient, and they are unique to the patient. So they were not inherited, they just happened by random chance. So in 2021, we summarized and compiled all of these progress and information that had occurred since in the last decade of genomics and their application to rare diseases, and we published this book, Genomics of Rare Diseases. And this approach of implementing genomic sequencing technologies to study human diseases, and especially monogenic diseases, has been so successful that now we have seen that certain countries, high-income countries, especially the UK, the US, and several countries in Europe, have started to perform pilot projects on how to implement genomic sequencing in newborns. So to sequence the genomes of children that had just been born, that many of them might not have any sign of a disease, but trying to identify at that point very early on if these babies are at risk of developing any condition, especially conditions that are of medical relevance when they are very little, when you can intervene very early and avoid unwanted consequences from these conditions that would result in disability and morbidity for these children. So in the same sense as we use newborn screening for neonates currently using tandem mass spectrometry. So currently several countries are exploring the application of genomic sequencing for newborn screening in the general population. So I mentioned a few of these points before. Why it's important to have an early and accurate molecular diagnosis. So I mentioned we can have early interventions to avoid adverse outcomes for children or patients at risk. Also, it can enable access to effective and personalized therapies designed to address the molecular defect specific to the condition. It allows for families and patients to plan for the future based on the natural history of the disease and what we know about the natural disease prognosis. We can provide accurate answers for parents and families. Also habilitates the genetic counseling process for family planning in case, for example, families want to have a second child and if there is a risk of genetic disease in that family. And ultimately, we can also use these to develop and implement public health policies based on disease prevalence that is specific to the countries of interest, ultimately reaching this goal of implementation of precision medicine. So genomics has really brought a shift in the paradigm of how we diagnose diseases. So I mentioned before the impact that it has had on identifying new disease genes and researching diseases, but also on the clinical management of patients with genetic conditions. It can be very impactful. So while we have currently the traditional paradigm of having a clinical diagnosis, where this paradigm focuses on the clinical signs and symptoms that are manifested by patients at a particular time. So the patient has develops a seizure, or develops some pain or some problem in a certain organ or part of their body, so they go see the physician. And at this point, then the physician evaluates this clinical presentation. And this can be subjective because it will depend on the knowledge and expertise and experience of the clinician. So if the clinician hasn't seen this particular symptom, this particular clinical presentation, they might not know what to do with the patient. It generally involves many times inconclusive tests that can take a long time to reach a diagnosis. And ultimately, it's a reactive approach because, as I mentioned, it starts when the patient goes and sees the physician and says, like, I have this problem. So it's a problem that's already there. It treats the symptoms that are already present in the patient. So this shift in this paradigm of having an early molecular diagnosis and even, as I mentioned before, starting in the newborn period before these diseases present, is an emerging paradigm that relies on the analysis of genetic and genomic information of the patient. It can be more objective because it's independent of the clinical knowledge of the physician. So even if the physician has never seen this particular condition, we know that certain variants in the genes, in particular genes, will cause definitely certain genetic conditions. So we can use also one test potentially to identify the diagnosis of the patient and in a very rapid and accurate fashion. And also, most importantly, it's a preventive approach. So again, it can identify diseases and predict the symptoms that will develop even before they manifest. So in some cases, this allows us to intervene early on and prevent some of these events that can potentially lead to long-term disability. So for example, in the case of what we call inborn errors of metabolism, which are genetic conditions due to abnormalities in enzymes that process different metabolites. So sometimes these metabolites start to accumulate, and the defect is obviously there from birth, but it starts accumulating, right? And then at some point, the patients will have what we call this metabolic crisis. And if the patient develops a metabolic crisis, generally these are very devastating crisis. They have neurological damage, and ultimately the patients end up with intellectual disability and affectation that will last for the rest of their lives. So if we know that these patients have these inborn errors of metabolism, sometimes we can intervene, we can change diet, we can manage what food they are intaking, what their ratios, for example, of amino acids that they are taking, and manage the biochemistry of the processing of these metabolites and avoid this metabolic crisis. And these patients that are handled appropriately before they have a metabolic crisis can go on and live healthy lives with adjustments to their diet, but without any major disability. So this is extremely important, and why an early molecular diagnosis is so important. Unfortunately, in Mexico, we have little access to molecular technologies, and the implementation of genomic testing and just broadly molecular testing for patients living with genetic conditions is very limited. So in 2020, we started this project, this initiative called the Mexican Network for Rare Diseases, Red Mexicana de Enfermedades Raras, which is a collaborative network of clinicians, researchers, patient organizations, and patient representatives that we are working on rare and undiagnosed diseases. And our main focus is to try to enable access, especially for patients with low economical resources, to genomic sequencing and other molecular testing technologies that can provide these accurate diagnoses for these patients. Also, we develop projects for education and outreach purposes to improve the knowledge and the awareness about rare diseases in Mexico. So in 2022, we launched the Rare and Diagnosed Diseases Genomic Sequencing Program. Here we get patients that are referred to us by medical geneticists that evaluate these patients. And then if they have a condition suspected to have a genetic etiology, so they are sent to us. We perform trio whole exome sequencing and perform all the analysis at the LIG here in Querétaro, in the Campus Juriquilla of UNAM. And then we do all the follow-up as well here. So from the first patients that we recruited into the study since January 2022, we have had about 30% of the patients with molecular diagnosis based on known genes that have already been reported. And we have about 40% of cases that have novel candidate genes that haven't been reported in the literature. Also, we launched the Rare Disease Patient Registry in February of 2022. And we also organized an annual International Rare Disease Day Symposium on Rare Disease Day. Ours was yesterday on February 29, to commemorate Rare Disease Day as part of the international efforts to raise awareness about these conditions. We also produce educational materials. So for example, for March, this is what we are releasing today to start, again, to educate and raise awareness about conditions due to trisomies. So we produce these infographics every month to talk about different rare genetic conditions. And again, as I mentioned, yesterday we celebrated Rare Disease Day, and we had the symposium and several activities. And if you're interested, you can follow us on social media. And also, this is our website, enfermedadesraras.lig.unam.mx. So this is one example of one of the patients that we have included in our study. So this is a Mexican 5-year-old male child from Sonora. This patient was referred to us due to autism spectrum disorder, developmental delay, sleep disorder, speech delay, and circadian rhythm disorder, meaning that he doesn't sleep when he should and he's awake when he shouldn't. So he also has a normal bilateral hearing, meaning that his speech delay is not a consequence of having a hearing defect. So he can hear normally, but he has been having issues with developing his speech. And also you can see that he has some frontal bulging. He has a bit of a bit of a little bigger head maybe than a 5-year-old his age. So we call this relative macrocephaly. He had previous testing, a normal karyotype, and an informative multi-gene panel. And when we performed the trio sequencing for this patient, what we found was a de novo mutation in this gene, ERF, which stands for ETS2 repressor factor. So this gene has been associated previously. It was reported in 2013 as causing this craniosynostosis type 4 condition. This is a transcription repressor that binds to different genes, including the ETS2 proto-oncogene, to repress the transcription of several genes, and it's involved in developmental apoptosis processes in the cell. So as I mentioned, in 2013, this paper came up describing 12 families with autosomal dominant and de novo mutations in this gene, causing heterogeneous craniosynostosis, and also craniofacial dysmorphism, language delay, and learning difficulties and hyperactivity. So 80% of the patients reported had learning difficulties, and especially with language acquisition, which, as I mentioned, this was one of the major concerns with our patient. Also, they had some particular features, facial features, including these frontal bossing and a prominent forehead and prominent eyes or orbits. So also in this paper, they reported some functional modeling studies using mice where they had some mice deficient with this gene. And you can see compared to the mice that have one copy of the gene. So in these two that are the mutant mice, you can see that the sutures, the cranial sutures, have started to close prematurely. This is how they should look. However, so this condition of craniosynostosis is exactly that, the premature closing of the cranial sutures. And also you can see that the cranium is shortened, is a bit more rounded, which you can also see here in this lateral view of the mutant mice versus the mice that have a functional copy of the gene. So this phenotype was consistent with our patient. However, as I mentioned to you when I described the phenotype of the patient, ours didn't have a diagnosis of craniosynostosis. However, based on this molecular diagnosis, we went back to the geneticist and we asked her to refer the patient to the neurologist to have an evaluation for craniosynostosis. And effectively, you can see here, this is the computerized tomography of the patient. So you can see that his sutures have started to close prematurely. So he does have a phenotype of craniosynostosis consistent with the molecular defect that we identified. Also here you can see the confirmation of the de novo mutation. So this deletion of 2 base pairs that we see in the ERF gene that occurred and is present only in the patient, and you cannot see in either of the parents here. So obviously we are doing these projects and these are important to enable these molecular diagnosis to patients. However, these efforts, although significant, they are small as in the greater context of the genomic programs and projects that are happening in the world. So there is very little representation of several populations around the world, especially as you can see here, what is generally known as the Global South. So populations in Africa, in Latin America are widely underrepresented, Southeast Asia, and they are very diverse populations characterized by admixture from indigenous groups and other groups. And in Africa, we know that there is a lot of genetic variation that hasn't been characterized. So we know that in genomics, we have a huge problem with diversity and representation of world populations. So you can see here the breakdown of participants per ancestry for different ancestries here, and 94.74 of the participants in genomic projects are of European ancestry. And you can see here the contribution of Hispanic and Latin American individuals, which is less than 0.5%. So this definitely has implications for public health and precision health, because this increases the disparities in access and benefits of genomic discoveries for underrepresented and underserved populations. There is also a lack of transferability of association markers and results from big genomic association studies to other populations that are not included in these studies. And there's also a lack of specificity for genomic interpretation of rare variants in these populations that may be or may not be medically relevant, but given the lack of representation and information about the frequency in these underrepresented populations, then we don't know how to interpret these variants. So there's a need to increase the diversity of genomic studies, but we have to do this in ethical, respectful, and inclusive ways for the populations where these participants are coming from. So I'm going to skip this slide, but basically these are examples of why it's important to increase diversity. Again, the performance of the prediction of certain genetic markers, in this case, what we call polygenic risk scores, while for European populations is very, very accurate, because we have a lot of information, as I showed you before, for European ancestry individuals. So it decreases very steeply for other ancestries and other populations that are underrepresented. So we discussed these challenges in this paper that came out, I think, 2 years ago, about the challenges of polygenic scores and the application in other underrepresented populations. So broadly, genomics has been recognized as a major field and technologies that can influence health and can contribute to reduce health disparities in the world. However, we are also at risk of increasing health disparities if genomic technologies are only accessible to high-income countries or individuals in resource in plentiful in countries with plentiful resources, again, such as the US, Canada, the UK, Europe, Australia, where they are able to do these large-scale genomics projects, but in other resource-limited countries, low and middle-income countries, we are not able to do this because of the high cost of the technologies. So the WHO has recognized this problem, and last year they published this report on accelerating access to genomics for global health and the need to implement genomics programs across the world to bridge these gaps in health implementation. And I was part of the working group that and the discussion tables about these applications of genomics as part of the WHO Science Council. So we have to have these projects be done in the appropriate settings with as part of collaborative projects, and not just sort of sending out samples out to be sequenced in the United States or in Europe, but actually have the involvement of local investigators, local clinicians, the strategic funding from diverse funders, build up the infrastructure both from a technological perspective, but also from a human capacity perspective, build that capacity, and then form these global consortia that are, again, truly collaborative efforts across the partners from high-income countries and also low and middle-income countries. So some examples of what can be achieved if we start having implementation of these larger scale genomics projects is the investigation of medically actionable variation in the populations of interest. So in 2013, the American College of Medical Genetics and Genomics of the United States published a list of recommendations of genes associated with genetic conditions that are of high penetrance and that have medically actionable interventions, meaning there are treatments or prevention guidelines or tests or screenings that can be done to reduce morbidity and mortality due to these conditions. So the majority of genes are associated with cancer, hereditary cancer syndromes, such as breast cancer, Li-Fraumeni, Lynch syndrome, etc. But also there are genes included for cardiac and cardiovascular conditions. So this list has been reviewed and improved over the years. Currently, we are in the version 3.0 that includes 73 genes. And again, this is a baseline, right? The minimum agreed upon list of genes that have potential interventions that can actually change the course of or prevent disease in individuals from population. We're not talking now about rare diseases like pediatric patients with rare diseases, but we're talking about large populations. So another thing that's important to mention is that these variant spectrum and frequencies can differ among populations. And so when we look at different large-scale genomics projects and look at the frequency and prevalence of variants in these genes, so we find that between 2 to 4% of participants from, again, the general population will have variants in medically actionable genes. However, as I mentioned, these can change between different populations. So we have seen that in certain populations that are that have a demographic history consistent with founding effects, such as, for example, the Amish or the Mennonites in Lancaster County, Pennsylvania, in the U.S., that are known to be founder populations, meaning that they started with the establishment of a very small group of immigrants from Europe that came to Pennsylvania, established their colonies there, and since there they have grown. So this the genetic pool was reduced due to that migration event, and then it was expanded once they settled and they started to build up their communities. So this causes what we call founder effects, meaning that if one of these original settlers had a particular pathogenic variant, when these people started having offspring and they started growing the families, so when it started with one person carrying this particular variant associated with a disease, now we see that that variant can expand and affect many people in that particular population because they are all derived from that original individual. So in founder populations, what we have seen is that the number of variants is much smaller than what we see in larger outbreed populations. However, because of these founder effects, some variants can rise to very high frequencies. So for example, in this case, in the Amish population, we have a founder mutation in the ApoB gene associated with familial hypercholesterolemia that is at 0.13, so 13% frequency in the population. So overall, we see that 14% of the population, of the Amish population, have a variant, mostly driven by this ApoB variant, of clinical significance in one of these 78 genes of the ACMG list. So again, just the difference between 2 to 4% in an outbred population versus 14% because of these founder effects. So the point of this is that Each population is unique, has its own sociodemographic history, and that has shaped the frequency spectrum of pathogenic variation in that population. So this is why it's so important to look at and do these genomic studies in the specific populations that are of interest, that we live in, that we want to benefit, because we cannot rely on borrowing frequencies and borrowing variants from other populations, because the variants in Mexican population will be different from the Amish and will be different from the UK. So this is why representation is so important, and inclusion of these other populations that are underrepresented in genomic studies is so relevant. So fortunately, we're starting to have advances and projects on large-scale genomics in Mexico, and this was published last year on October 2023 on 2 large projects on Mexican genomics. And so we're working with data from these projects. One of them is the Mexico City Perspective Study. This is the largest genomics study in a non-European population, so not just in Mexico, in Latin America, but also from a non-European population. So it encompasses 150,000 individuals from Mexico City. These include self-reported medical history, anthropometrics, and metabolic traits. And these individuals have been sequenced. They were sequenced between 2019 and 2021. So this data set includes 141,000 exomes and 9,950 whole genomes. And initial analysis on this data have identified 142 million genomic variants, with 1.4 million variants that are unique to this data set of Mexico City Perspective. Again, so these variants haven't been seen in Europeans, in populations from the US. They are unique to Mexico City individuals, and probably, obviously, to other people in Mexico. But again, because this is the largest data set that we have for Mexican individuals, this is very relevant. So initial analysis by us from the data that was publicly released in 2023 of aggregated frequencies data shows us that about 4.6% of individuals in the cohort carry a medically actionable variant. So in the genes that I mentioned to you before, and what we have seen in other populations, 2 to 4% of individuals will have a variant of medical relevance. So in the Mexico City Perspective study, we are identifying 4.6%. So this means that there are several highly penetrant actionable variants in the Mexican population that could potentially be interesting for developing screening programs for the population and guidelines to identify individuals at risk for developing, for example, breast cancer or colon cancer in the Mexican population. Also, this data has been already used and leveraged to identify novel drug targets by Regeneron Pharmaceuticals and AstraZeneca, including the drug targets for obesity and diabetes and other several that are currently in press. So we are performing all of these studies as part of collaborative projects in the region with the Collaborative for Rare Diseases in Latin America and the Caribbean, but also internationally with other organizations like G2MC, the UDNI, and the WHO Collaborative Network for Rare Diseases, to increase the diagnosis, research, education, and training on rare diseases, and more generally on precision health. So I hope that I have left you with a few messages today. So first, that rare diseases are individually rare, but as a group, they are very common and they affect a large proportion of the population. And you probably, every one of us knows someone that is living with a rare condition, and it's important to know more about these conditions because they have an important social and economical impact. Also, that enabling access to genomic sequencing for patients with suspected genetic disorders is of essence to have an accurate, timely, and definitive diagnosis that can help with disease management and treatment. However, we have issues and challenges with the cost and availability of these technologies in countries like Mexico and other low and middle income countries. So research programs can help enable access, but we also need to develop in conjunction with health authorities and government long-term initiatives for the investment and resource building for these large-scale projects and long-term projects to be sustainable at the country level. It is important to increase the diversity and representation of understudied populations in genomics, sorry, to achieve genomic equity for population health. So we have some cohorts for Mexican population that are becoming available, but there are still disparities that we need to overcome regarding collaboration, data access, and resource availability for sequencing other cohorts and building biobanks that can be informative for our population. And also, this is important because analysis of these large-scale genomic data sets can identify variants that are actionable medically in the population of interest, in this case, in our Mexican population. So with that, I'll thank the patients and the families that we work with, our collaborators in Mexico and abroad, and also the organizations that we work as well. And I'll be happy to take any questions that you may have.
Very well, guys, questions? Yes, go ahead.
Hello. Can mitochondrial diseases be detected by this genomic analysis?
Yes, but if you have a very strong suspicion that it's a mitochondrial disorder, so you might want to start with doing mitochondrial sequencing first, since it's a smaller genome, so it can be sequenced at very high depth and a much smaller cost. So you might want to start there. But since we also have mitochondrial disorders due to mutations in nuclear genes, so those we can see with these approaches as well.
Any other questions? Yes. Again.
Is there a limit to which type of diseases can be detected by this method? So, for example, neuromuscular diseases such as myopathies, could they be detected by this method?
Yes, of course they can. So in principle, there's no restriction, let's say, for any condition, as long as it's suspected to have a genetic etiology. The truth is also that not in all cases we detect a mutation or a known molecular cause. And that can be sometimes because the gene associated hasn't been reported yet, so it's not yet in the literature, and that's where all the research part comes in, because we need to gather enough evidence if we want to show that a new gene is potentially associated with a condition. So we still have a lot of genes that haven't been reported. Again, I said so far there are a bit more than 7,000 rare disorders that have been described in the literature, but we suspect that there are about 10,000. So we have about 3,000 conditions that are sometimes in people's laboratories waiting to finish experiments and be published, and some others that we haven't even gotten the patients and studied them. So there's still very many genes that we haven't yet reported. And in some other conditions, so for example, exome sequencing might not be the technology that will identify the variant, because maybe you have a variant in a non-coding region. So that's why, especially in high-income countries like the US and the UK, a lot of these is moving now to do whole genome sequencing as the ultimate test to diagnose genetic conditions, because then you have coverage of the whole genome and you can see structural variants, simple variants, and variants in non-coding regions as well. So in our case, because the cost of doing whole genome sequencing would still be extremely high for us, so we are doing first exome sequencing, and then in the cases that are negative for exome sequencing, which usually is about a 60% rate, a success rate. So in 60% of the patients where we do exome sequencing, we would identify the genetic cause. And then for the remaining cases, then we would reflex those to whole genome sequencing.
We have another question, please.
What do you like most about doing this kind of research?
So there are many reasons why I enjoy what I do and the research that I do. From a scientific standpoint, I think that, as researchers, we are driven a lot by curiosity and sort of there's quite a bit of excitement in sort of seeing for the first time a new gene associated with a condition that no one has figured out before. So I have myself identified novel disease genes for about 30 conditions. So that's one exciting part of it. Obviously, these translates into a practical application of being able to deliver a result to a family, to a patient that has this condition, and giving them a final answer to many times years of waiting and having tests and visiting different specialists to find what's wrong with them. So then having that possibility of impacting someone's life and giving them an answer. So for many conditions, as I said, just only 5% have an available treatment. So for many conditions, we might not be able to do anything medically. For some, we are able to guide better clinical management, not cure them, obviously, but improve the quality of life of patients, but also not just knowing why they have that disease. It's very important for families. So I think from the scientific standpoint, is very rewarding, sort of contributing to research in that way, but also from a social impact standpoint, it's extremely rewarding to be able to provide answers and diagnosis and help patients in their journey with these genetic diseases.
Also, another question, how do you overcome the hardships that come with the research, like mentally and emotionally? Like when you reach a roadblock in the research, how do you deal with it?
Well, you probably feel sad about it for a day, and then you go back and rewrite the grant or move on and rewrite the paper or incorporate the changes that the reviewer said that your paper was missing, and then move on and submit it to the next journal and do what's the next thing to do. So I think in scientific careers, you have to have a lot of resilience and drive, because yes, you do need to find ways to pull yourself up. It is not easy, an easy career, right? And it's subjected to a lot of criticism, right? From reviewers for your papers, for your grants, for this or that award or whatever. So there's a lot of criticism and judgment that comes with the scientific career, which is also part of the rigor of being a scientist. But you do need to find a way to deal with that in a constructive way, and pull yourself up when things don't go in the best way.
Well, we need to start setting up for the next talk, but we thank you very much for your interesting talk, Dr. Gonzaga Jáuregui. So a final round of applause for.
Thank you for the invitation, and looking forward to visiting you in person sometime in the future.
Of course.
Bye-bye.